All right. Hello, welcome everybody to this webinar. It is an absolute pleasure to be with you today. My name is Ben Liberson from Ebba Biotech. Now, we've got a nice participant list here. Question is, can you hear me okay? Are you able to ask questions? Can you interact with us throughout this webinar? I just want to check that before we kick off. Could you please drop me a note in the chat letting me know who you are and where you are listening from so that we can just check that all the tech is working fine. I can see people coming in already. Thank you very much indeed. Lovely to see you. As I said, it's fantastic to have you here. So here we are at the webinar from Ebba Biotech. We're a Swedish company started in 2015, and the core of what we do is interdisciplinary collaboration. You're going to hear a lot about that today. We have our product range, the OPTIR tracers, and we'll talk a little bit more about this at the end, but what I'd really encourage you to do if you're new or if you haven't heard about our new products for a while, that you get in touch with us, because our product range is actually updated with some really brand new formulations to make using them much easier for you. Okay, so without further ado, it is my pleasure to welcome our guest today, Oxana Klementieva, who is an Assistant Professor in Molecular Imaging at Lund University, and she is studying protein aggregation related to Alzheimer's and Parkinson's disease. These are topics that are very dear to our hearts at Ebba Biotech. And so without further ado, I will hand the floor over to you, Oxana. Welcome.
Okay, perfect. So I'm very happy to be invited by Ebba Biotech to present my work and to explain all of you how I use amyloid tracker to study amyloids directly in tissue. Specifically, I would like to talk about my two recent papers that we published this year. And in both of them, we used Amytracker, and I will try to explain why we did that and what benefit we had. So I'm Oxana Klementieva. I did my Ph.D. in Barcelona and then moved to Lund and become Associate Professor in Molecular Imaging. I also am Head of Medical Micro-spectroscopy Lab, and I hope that some potential master students could listen this webinar and could contact me afterward to do exciting work I could offer. So I'm at Lund, and I'm working at Nano Lund and Multipark, and also at Links. So in saying that, I would like to focus on research part. So a research topic of my group, Medical Micro-spectroscopy, is amyloids related to neurodegenerative diseases. And one of them are Alzheimer disease. So it is very well established that amyloid proteins such amyloid beta, tau, or alpha-synuclein can fold and form structural polymorphs. However, still we don't know which structure and which size of those structural polymorph can damage tissue. And since many years been in focus, we still have question which amyloid structure is neurotoxic and why protein amyloid begin to aggregate. And why we need that? If we could establish neurotoxic structure of amyloids, we could use this information to develop new tools for diagnostic and, of course, for effective treatments. So, to address those research questions, I use infrared microspectroscopy. And here in Lund, we have different modalities that ranging from micro to nano. Specifically, we have standard infrared spectroscopy, FTIR. Also, we have OPTIR that allows to study molecular structure at a resolution that is less than 1 micron. And also, we will soon have a new instrument that will allow nano resolution. So, and right now, OPTIR, optical photothermal infrared spectroscopy, is the main instrument that was used in my research. So to give a little bit background about infrared spectroscopy, I would like to say that infrared spectroscopy is a very established tool, and this tool can provide various information. It's like omics. Using this technique, we can get simultaneously information about proteins, about lipids, about DNA, about metabolites. But my interest is beta sheet formation. So, and this technique is extremely sensitive for beta sheet structures. And what is important, infrared can detect specific feature of beta sheet that discriminate beta sheet from other protein structure. And here, for example, I show yeah. Oh, sorry. And in this sample, I show proteins that, I show cells that grow directly on calcium fluoride, and those neurons express amyloid beta proteins, and that can be detected. And with study that we published in 2017, we discriminated early amyloid beta sheet formation that happen intraneuronally and can be detected by infrared spectroscopy. Later, my PhD student, Agnes Paulus, did very interesting work to look on beta sheet structures that come from different amyloid proteins like tau and alpha-synuclein. And what was really interesting that infrared spectroscopy can discriminate those proteins directly in tissue. So, however, this technique is diffraction limited, and maximum resolution that we can achieve is about 5 micrometers, and that makes very challenging to use this technique for subcellular study. So new developments has been done, and new microscope has been introduced to the market. So and this instrument called OPTIR, and that means optical photothermal infrared spectroscopy, that can break diffraction limit, specifically to overcome spatial resolution of to improve spatial resolution. This instrument use green laser and alteration. So we still use infrared to cause thermal expansion of a sample. However, instead of detecting infrared, we detect scattering light of green laser, and that makes spatial resolution constant and makes it 500 nanometer, depending on the type of laser used. So in our case, we use green laser because it's less toxic for tissue. So having that, we achieve sub-micron resolution, and that opens us extremely novel opportunities. So first of all, this technique is a non-contact technique. It means that we do not touch sample. And another very important thing, that this technique is taken in reflection mode. It means that we do not anymore need specific sample support that was required for Fourier transform spectroscopy. And that means that if we can use different sample support, then we could correlate different microscopy techniques applied on the same sample. And what is the most valuable for me personally, that OPTIR resolution is very similar to resolution that we can achieve using confocal microscope, and that allows to complementary studies. So, and how how it works. So first, we excite sample with infrared pulse and cause thermal expansion, and then we shine visible light, and with resolution of 0.5 micron, and then green light starts to scatter, and that scattering light we detect. And this scattering light very much sensitive to thermal expansion. And that's in this way we know which group is in resonance. So using this technique, we were looking on different samples, mainly on primary neuron that were directly grown, in this case on gold substrate, because the idea was to combine study with atomic force microscopy. So, and you could see image above. So, and then using the same sample, we could take spectra, and those spectra were taken at with the step of 0.5 microns, so really below micron resolution. And this spectra, for example, immediately we could see protein distribution along the neurites. So also with this technique, we were able to look on neuronal spines. So neuronal spines, this is a small protrusion from neuronal membrane, approximately 1 to 2 micrometers, but they're incredibly important for memory formation. And with this technique, we could observe that in transgenic neurons in APP-PS1 mouse model, so that could be important, we do not see aggregated proteins in spines, which may indicate that protein aggregation happens along the dendrite or axons, but not in spines. So it was very productive years. Since 2020, we published several papers. However, we had very difficult time to reply reviewers' question, what exactly you're measuring? Cells or tissue are very heterogeneous. Can you prove that you detect amyloid beta? And we had to do a complementary study using immunofluorescence with a lot of sample, trying to show that yes, what we measure is exactly amyloid protein. So, and then using Amytracker and using a novel setup that become available this year. So last year we published, we did 2 exciting projects and published them. And here I would like to focus on those 2 publications and present them a little bit more in detail. So using for these 2 projects, we used fluorescently guided microscopy. As a fluorescent guide, we used Amytracker, and that helped us a lot to find our protein of interest and measure protein structure. And of course, using Amytracker, we labeled amyloid beta protein in tissue and then we look at the microscope, and we found very fast our amyloid plaques and guide our spectroscopic measurements. And it was extremely exciting. Those 2 key modifications allowed us fast targeting. We could find amyloid plaques, and we could retrieve immediately structural information. For example, in that image, that if you could see my cursor in that one, you could see amyloid beta labeled with Amytracker and microglia that is attacking Amytracker. And we could take specifically spectra from those amyloid aggregate and from the cell that is attacking amyloid aggregates. So how this work? We use green laser to detect photothermal expansion, and also to detect fluorescence. So we added one more. Having inbuilt already green laser, we just added detector for fluorescence detection, and we got two readouts from the same spot immediately. So, and for us was extremely important that colocalization of fluorescence and spectra was exact. So, and here is beautiful brain tissue of APP presenilin mice that develop a lot of amyloid plaques, and you could see them here labeled with Amytracker as the white dots, and you could see how Amytracker zoom in fluorescence, and then in the right image you could see how look amyloid plaque in infrared. So, and the most important for us that using sub-micron resolution of approach, we could assess amyloid structure from corona, from core, and from tissue that doesn't have amyloids. And of course, it was we spent some time to characterize the spectra of Amytracker, and you could see that it's not overlapping with the part that gives us information about beta sheet. So actually, this is ideal dye to use for infrared spectroscopy. So, and here you could see how the example how it can be done. So we detect amyloid plaques in tissue, and then we take spectra with a really tiny step of that allows confocal platform with 0.1 micron, and we could characterize beta sheet in at subcellular resolution. So, and the most important thing was that using Amytracker, using fluorescence guidance, it was very easy to label tissue. It was good fluorescence, and it made our measurements incredibly faster. So, and here I could demonstrate the possibility of technique of infrared spectroscopy to detect amyloid aggregates. So here is distribution of beta sheet shown as a red marker, and I measured the distance, how big those aggregates can be detected. And it is extremely important that we could see structural heterogeneity. So the green is less beta sheet, red is a lot beta sheet, yellow is moderate. So, and you could see that you could see structural distribution of structural information. And using dye, we could say, we could confirm that this structural information comes from specific spots that are positive to Amytracker. So, and of course, this technique allows us to take single spectra from single aggregates and assess structural polymorphism that we could observe in tissue. So, and the ability to use Amytracker, we made next step, and we did very extremely exciting project. So what we did, we took acute brain slices, and those brain slices are still alive for a few hours, even brain is cut, and we use Amytracker to detect amyloid plaque in living brain tissue, and then we could look for the first time on amyloid structures that are in hydrated tissue, and we think still in living brain slices. So, and how it looked like? So we took brain tissue, and we did first images infrared. We used Amytracker to locate very fast amyloid plaques, and then we took spectroscopic images, and we took time point 1, and then we imaged in half an hour another the same time, the same brain area. And what we observed that we could detect newly formed amyloid, but not only amyloids, so the red spots, it's a beta sheet structures. We could see also new structural features that correspond to lipid oxidation. And that information can be retrieved simultaneously, which make this technique very valuable for amyloid research. So, and here's a little bit process. So the brain on the image, on the left image, is cut on vibrotome. So the brain is just extracted from the animal, and vibrotome cuts quite thick section, about 300 micrometer, but that thickness let brain tissue be alive. We placed it in the buffer, and in that case, since we started developing support that allows to prolong measurements on hydrated tissue, but at that time we just put it in Petri dish with just a little bit of buffer, and then we just make sure that the objective doesn't collide with Petri dish, and then we did a spectroscopic images. And here, just an example, if you look on the bright field, it is incredibly difficult to find amyloid plaques. But so you need to zoom, and then maybe those black densities will be amyloid plaque, but not necessarily. So, but using Amytracker and labeling amyloid plaques, it becomes really handy because you do not need to screen tissue and look on spectra if we have shoulder that corresponds to beta sheet structures or not. We could find it, and we could zoom immediately to the interesting spot. And after that, it was also important to have Amytracker because it allowed us to make electrophysiology on the same spot that we did spectroscopic measurements. And it was absolute surprise that we could find still living neurons in the tissue that was subjected infrared spectroscopy. So, meaning that we could extend our windows for spectroscopic measurements and to assess amyloid structures in living tissue. So, and that is our next project that we're working currently. So, and here I could show that we observe different plaques in brain tissue and compare them with plaques that were in fixed tissue. And that was also the part of the project. But here I just want to highlight that super resolution infrared combined with Amytracker imaging is an ideal tool that can go beyond of perception of just infrared spectroscopy and provide more information. And using Amytracker as a guide for OPTIR measurements to assess amyloid structures in heterogeneous sample like neuron or tissue makes possible to makes study much more robust and much more faster. So this is my group. We are 11 people right now. We collaborate with Lund University, with other group at Lund University, and we have a lot of interesting projects. So, and I hope I shared my enthusiasm about those projects. So, and with that, I will be happy to take questions.